All right, guys, Mr. Antonucci, and we're back here. And in this video, we want to talk about how to determine when it's appropriate to use paired T procedures versus when it's appropriate to use two sample T procedures. Because up to this point, we've used two sample T procedures to perform inference about the difference between two population means, but we also used pair T procedures to perform inference about a population mean difference. Okay, so here's the thing. If you're presented with paired data in the AP Stats exam and you analyze them as if they were independent samples, then you're going to lose credit because the choice between paired T procedures and two sample T procedures depends on how the data were produced. So make sure you read your question carefully. And here's a couple more tips. If the data sets have unequal sizes, then they can't be paired. They had to come from two different samples. If the data table has a third row or column with labels for values in the other rows or columns, the data are likely paired. Questions about paired data typically use the phrase mean difference, whereas questions about two independent samples typically use the phrase difference in means. So it's very important to be able to distinguish based on how the problem is presented, whether we have paired data or two sample T procedures. So in this example here, we're going to decide whether or not to use paired T procedures or two sample T procedures to do inference and explain why in each one. So in this example here, before exiting the water, scuba divers removed their fins. A maker of scuba equipment advertises a new style of fins that is supposed to be faster to remove. A consumer advocacy group suspects that the time to remove the new fins may be no different than the time required to move the old fins. On average, 20 experienced scuba divers are recruited to test the new fins. Each diver flips a coin to determine if they will wear the new fin on the left foot and the old fin on the right, or vice versa. The time to remove each type of fin is recorded for every diver. So in this case, it's definitely going to be T procedures because the data come from two measurements of the same variable. In other words, the time to remove the fin. So it's the same person, but they have one fin on one foot and the other type of fin on another foot. So you're not doing two different samples in this case. Okay. Our next example. To study the health of aquatic life, scientists gathered a random sample of 60 white piranha fish from a tributary of the Amazon River during 1 year. The average length of these fish was compared to the average length of a random sample of 82 white piranha from the same tributary a decade ago. In this case, this would be a two sample T procedure because first off, you have two different sample sizes. One is 60, one is 82, and it's two different samples here. So the data come from independent random samples of white piranha in two different years. And our third example here, can a wetsuit deter shark attacks? A researcher has designed a new wetsuit with color variations that are suspected to deter shark attacks. To test this idea, the researcher fills two identical drums with bait and covers one in the standard black neoprene wetsuit and the other in the new suit. Over a period of 1 week, the researcher selects 16 two-hour time periods and randomly assigns 8 of them to the drum in the black wetsuit. The other 8 are assigned to the drum with the new suit. During each time period, the appropriate drum is submerged in waters that sharks frequent, and the number of times a shark bites the drum is recorded. Really long setup to that one, folks, but this is definitely a two sample T procedure. And this is because the data come from two groups in a randomized experiment, with each group consisting of 8 time periods in which the drum with a specific wetsuit, whether it was the standard or the new one, was randomly assigned to be submerged. All right, as we wrap this up, I want to just talk about a couple additional important tests, or excuse me, notes rather, about using tests widely, because significance tests are used widely in reporting results of research in many different fields. So one of the things that we have to be careful of is statistical significance is not the same thing as practical importance. So when a null hypothesis of no effect or no difference can be rejected at the usual significance level, there is convincing evidence of a difference. Okay. But that difference might be very small. When large samples are used, even a tiny deviation from the null hypothesis could be significant. And so, while a difference we might have convincing evidence of a difference, in all practicality, it might not really mean much of anything. And then the second thing is beware of multiple analysis. Statistical significance should mean that you have found convincing evidence of a difference that you were actually looking for. So the reasoning behind statistical significance works well if you decide what difference you are seeking, design a study to search for it, and use a significance test to weigh the evidence. In other settings, however, statistical significance might have little meaning. Okay, so that's it for this section, guys. Hope that was helpful to you. As always, reach out if you have any questions, and take care.